Okay, we're at the top of the hour. How about we get going here? Hi, everyone. Thanks for joining us today. My name is Ed Creedon. I'm a senior project manager with AST Environmental. And today's presentation is going to be given by Edward Winner. Ed is Vice President of Remediation Products, Inc., otherwise known as RPI. And Ed's presentation is going to run, I think, about 30 minutes, 40 minutes. Depends on how excited he gets as he's talking. But there's going to be plenty of time afterwards for some Q&A. And I'm guessing most folks on here are very familiar with this platform, but I do want to point out there's a question tab in your webinar control panel. So jump on there anytime throughout the webinar and post your question, and we will get to those once Ed wraps up the presentation. I'd like to just take a minute before we get that started. We still have some folks logging in, and I'll just take a moment to help you understand the relationship between AST Environmental and RPI. We are part of, AST is part of the RPI group. RPI group is made up of RPI, who is the developer of the activated carbon-based technologies known as BOS 100, BOS 200, 200 plus, and CAT 100. And then also in the group is the company that I work for, AST Environmental, and we are the third part of the group is the authorized installers. RPI is based out of Denver. AST is headquartered in Lexington, Kentucky area in Midway, actually. And there's a number of authorized installers around the United States, in Canada, Australia, and in Northern Europe. And our role at AST is the national and international project assessment and design. And we also are responsible for quality control of field operations. And we are the worldwide distributor of all of the RPI products. The authorized installers, it's a group of companies that have completed extensive training for field installation, and the many years of shared experience and acquired knowledge of this group is really a critical component of the overall success of the projects, because I think, as most of us know, the best designs are really only effective when the products are properly installed and they make contact with the targeted contaminant mass. So if you have interest in learning more about the field methods, we're not going to be talking about that today, of how we apply these technologies and get them where we need them to be in a variety of different geologic environments. I invite you to visit our website. That's astenv.com. And there's an extensive library of recorded webinars that they're excellent learning tools. You can view them at your convenience, and I hope you take some time to check that out. And then there's also a lot of information specific to the technologies on the RPI website. That website is trapandtreat.com. So I encourage you to go to both of those websites, actually. And there's really a lot of great information available for you there. Okay, so let's get this thing kicked over to Ed. His presentation today is going to help us gain a better understanding of the energy transfer process across activated carbon and why this is important to us as remediation practitioners. So I will be back at the end of Ed's presentation to lob some questions his way. And Ed, please take it away.
Okay, thank you. Can you hear me?
I've got you.
Excellent, excellent. So I like the title. It kind of hit me, your diet strategy for the new year. Every new year, people will try a new diet out, and it lasts, a month and a half. But what is key in almost all diets is an understanding of input to output. So I have a water wheel up on this screen, and that input is your food. It's what you're eating. And everything you eat ultimately is broken down to electrons that are transferred to air and blown out of your lungs. Okay, now some things go out as waste, but I'll spare you that. The idea is that all of life is a water wheel, that we take electrons from higher energy and take them to a lower energy. And in a lot of diets, what they tend to stress is, how can you use a lot more of those between the time you take them in and the time you get them out so that you don't store the excess as fat. This is true for every living creature. They all have to take in food. Ultimately, they pull electrons at higher energy, take them to lower energy, and get rid of it. So microbes are no different. And how well they metabolize whatever it is you want to get rid of or they want to eat depends on the system in which is in play. So what is direct interspecies electron transfer? And the rather smart alecky, why do I care? That's kind of odd. But anyway, what is diet? It stands for direct interspecies electron transfer. Some microbes can transfer electrons to other microbes without packaging them into intermediate chemicals. This is really big. It's maybe only been about 10 years ago that people found out that this could even occur. Before that, they always thought you had to have an intermediate. And it would look something like this, that you would break down some sort of organic substance to CO2 and hydrogen, and that hydrogen would float through the media to some other microbe, which would deal with the hydrogen in some manner. Ultimately, in an anaerobic environment, the hydrogen combines with CO2 and makes methane. But diet is a process in which you transfer that electron without the hydrogen ion. So if you look up in the upper right, you can do this by conductive pili. You can think of pili like flagella. You've seen microbes under a microscope and something's flopping around. You can think of them like that. They're kind of modified flagella in which they can attach to a conductive surface, or they can attach to another microbe and transfer electrons. If you look down below that picture, there are electron transporting proteins. And these proteins would allow the transfer of electrons directly. Or of course, what I'm going to stumble upon based on the fact that our products are activated carbon based is the picture on your right, which is the electrically conductive material. So this is a very complex picture, and I'll spend a couple minutes on it. But when you think of anaerobic degradation, and I'm going to use hydrocarbons throughout this particular presentation. We could talk about chlorinated solvents, but the pathways are a little different, and I don't want to mix them. The same pathways that are breaking down carbohydrates, proteins, and lipids through, we use hydrolysis as the general characterization of that, also hydrolyze hydrocarbons. So this is the entry of the breaking down of hydrocarbons into the biological process, hydrolysis. So you break it down and then you take those breakdown products and break them down again in acidogenesis into short chain fatty acids. Then acidogenesis, you'll break it down into acetic acid, hydrogen ions, and CO2. That's through the middle of this picture. Now, if you look at hydrolysis, the point of hydrolysis is to break big sugars down into small sugars, to break amino acids down into peptides, to break long chain fatty acids into smaller chain fatty acids, to break your hydrocarbons down into their subpieces by various means. There is a way in which you can push all of these reactions. If you think about it, in a chemical reaction, if you have more reactants, you get more products, right? There's kind of a push there. And the same thing can happen to some degree with microbes. So if you feed them more sugars, you'll get more short chain fatty acids, you'll get more hydrogen, you'll get more acetic acid, etc. Same thing with peptides. The same thing with hydrocarbons, except hydrocarbons have a little bit of a problem, and that the mixtures that you're dealing with, some of them are toxic to the microbes too, so they have to take them in. They have to sip, so to speak, right? But the Boss 200 Plus product that we have out, it pushes right here where that blue arrow is on your right. It pushes on that, what we're calling monomers here. And by pushing that, you accelerate the processes further down. Now there's a really interesting bleed off, and think of this as a bleeder valve or like a, on a steam pot, to release the pressure. If you look at acidogenesis, you'll see I've added a little extra arrow there that goes off to the right, and it says polyhydroxyl alkanoates. Now, this is a really interesting thing, has not much to do with diet, but has something to do with overall microbial metabolism. And that is, if you push where that blue arrow is by providing more of the things that are monomers, the microbes, which in the wild, okay, and that's what we're talking about. We're talking about subsurface. We're not talking about laboratories. We're talking about an open system, not a closed system. If you push, the microbes, which are normally starving, say, oh my gosh, look, there's all this food. And they start polymerizing that food into long chain polymers that they can store. How long can they store? In a biofilm, this can feed the biofilm members for years. And understand, for a microbe, years are like centuries, right? They don't live very long. So this is a really interesting bleeder valve. When you put in too much food, it doesn't choke them. They just start stacking away. Now, actually we do the same thing, right? When you eat a bunch of food, the food is stored as glycogen in your liver for when you don't have food. And so that's what they're doing. They're storing food for when they don't have food. Not all microbes can do that, but in biofilms, certain microbes seem to be assigned to hanging the hams in the barn. So that's an important bleed off. In the acidogenesis, it sounds so much like aceto, the next reaction. You make these short chain fatty acids, and then acetogenesis, you make acetic acid, hydrogen, and CO2. And this is where the making of the hydrogen is where the diet process comes into play. And you see that I have diet up to the right, and you see that I have diet lower in the process. Now, the hydrogen is really interesting because you want to push, you want to make hydrogen. A lot of reasons you want to make hydrogen. One reason is a lot of hydrogen can actually help you break down your hydrocarbons. The difficulty is if you make too high a level of hydrogen, it is just like similar to any other chemical reaction. If you have too much product, it slows the reaction, right? That's an easy way of thinking about it. The microbes don't look at it quite like that, but that is the end result. So diet, by directly transferring electrons to the next microbe, you can avoid hydrogen buildup. So like the polyhydroxyl alkanates, this is another bleed-off valve that keeps the reaction moving forward. So diet will not take over, except in laboratory experiments and published papers, where they manipulate the population. The population diet will not take over the entire electron transfer of the population. It'll only end up being, we're talking 6 to 10%, something like that of what happens, but that's enough to act as a bleed-off valve. It's very important. Another thing I want to show you, if you look to the far left of this screen, do you see the label hours to days, minutes to days, minutes to hours, seconds to minutes? The reason this is important is people sometimes don't have a good sense of how quick things are happening. Breaking down your hydrocarbons, breaking down fatty acids, this takes hours to days. Taking that and breaking it into short chain fatty acids takes minutes to days. Once you have those short chain fatty acids, and you're going to break it down into acetate or acetic acid, same thing, hydrogen ions and CO2, it takes minutes to hours. But to take that, either acetic acid or hydrogen and CO2, and channel it to methane takes seconds to minutes. So if you can increase the efficiency of actually methane production on the other side, which you can do with activated carbon, a lot of publications cover this, and you're bleeding it off in seconds to minutes, you actually will not build up high levels of methane. Now you'll see in some published studies, primarily bottle studies, and that's where you put everything in one bottle, a closed system, you'll see they build up a lot of hydrogen. And of course you do. It's a closed system. What do you expect? But nevertheless, let me move on to the next slide. Okay, so this just reminds you of what you do know about hydrocarbon degradation, that in general, hydrocarbons are broken down by a syntropy, means lots of microbes mixed together, not just one species. And they essentially will end up in an anaerobic degradation process to be hydrogen and acetate. The hydrogen and acetate can either dump electrons to nitrate, sulfate, iron, manganese. You've seen that before. Or they can break down the acetate to hydrogen ion and CO2 and make methane. And it is primarily this upper process that we're talking about right now as a bleeder valve to a larger biodegradation system. Throughout the degradation process, electrons are moved, just like that water wheel that I put at the first of the presentation. You've got to move electrons, whether you're doing fermentation, whether you're doing acetogenesis, you've got to move those electrons. Now, as I mentioned earlier, how people thought for many years this worked is that hydrogen and formate were the only way to transfer the electrons from a donating bacteria. So you'd break down these organics into acetate, CO2, and hydrogen, and then the donating bacteria would produce hydrogen or formate, hydrogen ion on the previous one, this is hydrogen, and formate to transfer to methanogens. But interspecies direct electron transfer between bacteria and methanogens can happen directly. They don't have to make those intermediates. This is big. This is really big. The efficiency is ninefold higher than transferring it by a chemical intermediate. Therefore, it happens quicker. If you provided a conductor and it's doing diet across a conductor, if those are the right microbes, then not only does it happen quicker, it happens much earlier in the degradation process. Why is this all important? If you don't have to make that intermediate hydrogen, then the bacteria itself doesn't have to produce the enzymes necessary to deal with making hydrogen or formate, at least not at the same level. And not requiring that carrier conductor is quicker. This overcomes thermodynamic limitations of high hydrogen partial pressure. Remember I said, you have reactants and products. In a sense, hydrogen is your product. If it gets too high, it feeds back on the earlier reaction. The electron transfer rate with diet itself can be 9 times faster than using those chemical intermediates. And this also allows something yet more interesting. You can actually degrade contaminants to a lower final concentration because it's thermodynamically more favorable. Now, this is a methanogenic biofilm. On this particular picture, this is magnetite. It's not activated carbon, though it's black. And what you see, I have 2 different microbes that have been stained. There are many more microbes here. They just chose to stain a key electron, the 2 key electron transfer microbes, right? And I will show you this here. So if you look at the bottom picture, where it looks like you've got hair on a rock or something, those are actually microbes on activated carbon. And one of the microbes is Geobacter metalloreductans, and the other one is Geobacter sulforeductants. So the metalloreductants gives the electron to the sulforeductants without using the intermediate. And this is a very well-known duo, so to speak, though other microbes are involved, on activated carbon. So why is the activated carbon aspect so important? I could just use magnetite, as you saw in the earlier picture. The activated carbon will absorb the hydrocarbons, lowering the concentration so they're not as toxic. It provides a structure on which microbes can live and find protection from predation, and they will build a thick and rich biofilm on that structure. That enriches the microbial population. Enrichment means you got a lot more of them. And then they will produce, at least to some degree, a diet as a bleeder valve if you're feeding them extra food, as we do in the BOSS 200 Plus. Now here, it's probably not the greatest example, but I was digging around and found this pretty quickly. These are, this is genomic sequencing from a BOSS 200 Plus site, and you will see in the middle picture, top in yellow, Geobacter metalloreductans, and then you go lower, you see Geobacter sulforeductans. This is the same group I just showed you in the previous slide, where the Geobacter metalloreductans transfers the electron directly to the Geobacter sulforeductans. So you can see it in our genomic sequences. Geobacter lovely was the first microbe examined for diet. And you will also see that you have Geobacter metalloreductans over on the left and that sequencing. And then the two on the far right, you see more Geobacter. You see methanotrex, which is another electron recipient from the Geobacter. And you see it similar below. And this is not really meant to prove anything. It's just to show you that, yes, we are seeing these kinds of relationships. The numbers are percentages. Since these are samples pulled from monitoring wells, you have to be careful about those percentages. What you will find is that the electron recipients usually are really entrenched in the biofilm, and you tend to see them in lower numbers in samples pulled from monitoring wells. Okay, this is one of those slides that they always tell you, don't show people stuff like this because it's too busy and it's too hard to understand and they don't get the message. But I need to show it to you anyway. And here's the reason. This is a sample from a bioreactor. Very tiny sample from a bioreactor that was sent, and they did genomic sequencing, and then they looked at the metabolic pathways that were being expressed. And what they saw were you've got fatty acid degradation happening at the very same time you have formate generation. Remember, the formate's carrying that hydrogen. You see a little bit of O2 respiration despite the fact that this is anaerobic. You see proteases breaking down proteins, you see amino acid degradation, you see sugar generation, you see hydrogen ion generation. And if they had looked at it in this particular study, you would have saw various levels of diet. Because what they did in this study is they took sludge, and they use sludge in the bioreactor, but the sludge is very similar to, a wild type. It's similar to the subsurface. It's not a laboratory creation. It's got lots and lots and lots of different microbes in it, and you find that they build very complex relationships. And those complex relationships are key to understanding how do you become successful in bioremediation. A lot of people want to sell you one microbe and say, this microbe will solve all your trouble. It will not. That microbe is like an animal that lives in a petting zoo. You put it out in the wild and it's dead in a heartbeat. Scaling is key to understanding what's happening in microbial life. It's actually, for any of you who have to be statisticians, you know that scaling is your biggest trouble in statistics, right? How strong is your input? But let me help you think about scaling of size for microbes. Let's say you're listening to this seminar and you're a typical American female. You're maybe about 5'3" in height. Then 1,000 times your height away is a mile away. For a bacteria, an inch is like 12.7 miles. So you can understand that in an area of half an inch, you can have so many different metabolic reactions occurring all at the same time, and they all be important. Yeah. Timing. Scaling is important, but timing is important. Most of your published papers look at the biological reactions as if hydrolysis occurs, and when hydrolysis is done, you go to acidogenesis, and when acidogenesis is done, you go to acetogenesis, and finally you end up doing nothing but methagenesis. That's all that happens. And the reason they see it that way is because in order to understand a thing, you have to put the least number of changing elements in it. So you look at 2 microbes in a bottle. But in the real subsurface, hydrolysis continues, and you need that because you're going to be biodegrading on your hydrocarbon sites for 18 months, 3 years, depends on what the concentration of contaminant lost is, right? The acetogenesis process is going to carry out. And methagenesis. The thing that carbon helps you do is start all of these processes earlier. And all of this has been published in multiple places. This is not questionable. And by doing that, you actually keep the methane level much lower. You keep the hydrolysis level much higher. And because you are actually processing things to food, you keep the microbial population richer. So specific location makes a difference. So scale, timing, and specific location. When you do make the hydrogen, what happens to it? Most of it ends up interacting with oxygen as it moves upward in the subsurface if it is not processed by other microbes. So you don't really have to worry about, and I need to make this clear because sometimes people say crazy stuff. You are not going to build up enough methane to blow up a site. Never, ever, ever. Doesn't happen. But in anaerobic degradation, you do make methane. So what's the summary here? Direct interspecies electron transfer. Some microbes can do this and they transfer electrons without first packaging the electrons as chemical intermediates such as hydrogen. Using activated carbon is one way some microbes can transfer electrons directly to other microbes, and they tend to act kind of as a bleeder valve in a larger system. This may improve the efficiency of biodegradation 9-fold, which is big. Diet is the wild part of a larger scheme of metabolic pathways that occur together in biofilms. So keep in mind that microbes live on their own scale, that when you take a sample out of a monitoring well and look at whether it's aerobic, anaerobic, whether you've got methane levels or whatever, it's completely different than what the microbes in the subsurface are seeing. And geobacter species can form these syntropic relationships with methanotrex as an example I showed you to function via diet. We see that in our BOSS products, BOSS 200 and BOSS 200 Plus. Enhancing the electron transfer with activated carbon stimulates the degradation of organic waste. And this we regularly see. And so that was really the large gist of what I wanted to present to you, to be able to see how that microbes have diverse metabolic pathways that they are pulling off all at the same time. And they, if in the wild, they're allowed to build up their own biofilms and their own systems, that they will have bleed off valves that keep them in balance. And that so much of what you read that comes out of our industry is really like building a petting zoo. But we are not doing that. In the subsurface, it's a wild world there, like the African savanna, and you got to have a lot of different species, and they have a lot of different metabolic pathways that are operating all at the same time. So your diet strategy, so to speak, has to be to eat a lot of different foods, move through the various possibilities, don't get stuck on one thing. Ed?
All right, thank you, Ed. That was excellent as always. I know I certainly learn something new every time I hear you speak about this. We do have just a couple questions, so if any of you out there are still mulling over some questions, fire away while we get to a couple here. The first one, Ed, just kind of a basic question that I think is a good one to help remind us what's going on here. How is BOSS 200 plus increasing biodegradation?
Okay, I smell a plant. I smell a plant here.
Uh-oh.
Nevertheless, let's see. Here we go. Okay, I brought back up onto the screen this earlier overly complex slide. And the way the BOSS 200 plus, at least part of the way that it's working, is by providing amino acids and nutrients. We're pushing the production of short chain fatty acids, which feed a larger, more diverse microbial population. That is key to what we're doing with BOSS 200 plus. And if you feed that population long enough, and we have a mixture that does, you will form deep, heavy biofilms in the subsurface, and they can sustain themselves. They can sustain themselves through seasonal changes. They can they can sustain themselves through changing concentrations of contaminants, much better than planktonic microbes. Planktonic means microbes that are just floating around, okay? And that's really a key aspect, not the whole thing, to what BOSS 200 plus is doing and how that pertains to this particular presentation. The other aspect is it has an activated carbon base, which we've been talking about all through here, which is a conductor. And by having that conductor, you give the microbes extra options for getting rid of electrons as opposed to just building up hydrogen and formate, which is energy expensive for the microbes. Ed?
Okay. Well, and you did spend some time talking about using activated carbon as a conductive material. So what are the other ways that the diet process occurs? Can you touch on that again?
Maybe. See if I can find it. Maybe hit it in the wrong direction. Let's go back up. There we go. Yeah, and this is really cool. So if you get a chance, I mean, you could read this like in Scientific America. You know, they have some good articles that are pretty general for the average reader. That you can carry those electrons across electrical conductive pili, and those pili are like flagella. You know, you've seen under a microscope the little flagella flopping around. Well, the pili are modified flagella, and the microbe can kind of come up, grab another microbe or a conductive surface, and send electrons across it. The electron transport proteins, these are really varied, but the one that you might be able to relate to as an audience is heme in your blood, right? There are hemins, which are on the surface of some microbes will use heme, and they can carry electrons and transfer it to another microbe. So these are other ways. And in fact, if you pull microbes from the wild and you get a pretty good population, what you'll find is multiple diet processes occurring simultaneously.
Okay. All right, we've got another question here. Could you explain how the microbial population shifts over time and how are new bacteria introduced to the system? Were they dormant?
Yeah, this is an interesting question because if you do genomic sequencing in the manner in which we do it now, you have what they call deeper and deeper runs. And I won't go into what that is, but you can see more and more microbes the longer, in a sense, you run the machine. Now, you start getting error the longer you go too. But I think most people have come to the conclusion now that there are lots of microbes that are dormant in the subsurface, and they, it's the starvation lifestyle they live, right? They're just hanging out, waiting for the day when it's appropriate for them to start reproducing. And so the depth of microbes that are available to you, even to degrade hydrocarbons, as an example, is probably unknown until you actually set up the environment that's appropriate for them. And simply having a hydrocarbon release is not in and of itself the appropriate environment. Again, the hydrocarbons are toxic. They lack certain nutrients. So yeah, if you stimulate the environment in some way, or the environment changes, in the fall you get in the spring you get root exudates, and if you look at your sites carefully, you'll see that you get more degradation with that. In the fall, when the leaves start degrading, and you get a little bit more bloom of microbes with that. So the microbial population does walk through the emphasis on which members are most active over time. So there are some microbes that just basically, they break down hydrocarbons really well and really fast. They just don't do very well with high concentrations. Others seem to do pretty well with high concentrations. So you might, for example, see a change in the microbial population in that manner as you walk through the time of the site. Also, if you're providing, as we do with our product, if you're providing electron acceptors, you will see the population change as the electron acceptors are used up. So I'm not sure exactly if that's what you were looking for, but hopefully that was helpful.
Okay. All right, got a couple more here. Does your BOSS 100 promote diet better than activated carbon without metallic iron?
Yeah, that in itself is almost a whole nother presentation. And
we've got time.
The BOSS 100 does promote diet, and we'll just take it from the sense as it promotes the exchange of electrons. It is very different. It's very different than what you would see for hydrocarbons. Hydrocarbons are much better understood. I have to say that a lot of what I'm seeing with the BOSS 100 and the CAT 100 are a little bit harder to understand and a little bit harder to express. And so I'm really still working on it, and I don't want to walk too far out on this plank and then find out that I've been sawing it off behind myself also. But yeah, you will see a tremendous number, especially the geobacter, just boom with the carbon that has the iron in it.
Okay.
Sorry, I didn't give you a more complex answer. I'm not really ready to do that right now.
No, that's good. Okay. Let's see. Would a broad spectrum bacteria culture facilitate diet process sooner, or do we have to wait for the system to build and equilibrate?
Yes and yes. If you provide the appropriate microbes and lots of them, you will build up that system sooner. So, we do provide microbes, and the microbes are not purely, the intent of the microbes is not purely to degrade hydrocarbons directly. So, we've kind of already figured this out, that microbes do more than just degrade hydrocarbons. They help to build systems and support systems. So if you think of it as a city, you got the tow truck driver, you got the milkman, you've got the, insurance sales guy, et cetera, right? So in order to build a complex system, you need a lot of different capacities in the microbes. And you can stimulate that by providing the microbes. And what you'll find is that initially the microbes you supply are pretty active, but over time most of them die back, and a few select ones, which vary from site to site, end up becoming, the new immigrants that everybody's accepted, so to speak, and have their little duties and their jobs and are doing well. So it kind of goes back to that idea, we're not really building a petting zoo. It's kind of a rough world out there. And yes, you can encourage it, but yes, the subsurface kind of bit has a mind of its own, and you have to kind of work between those two.
Okay. Getting more popping in here. I think you're stimulating some brains out there. So first of all, this questioner says, thank you for your presentation. Do the redox conditions of the site change after injection of the products? And there's a follow-up to that.
Yeah. Okay, so thank you. I appreciate the compliment. The redox does change. Okay, so when you first inject, and this is more of an AST kind of thing but when you inject carbon under pressure, high energy, you open up spaces and carbon goes into those spaces. In those spaces, you now have a better transfer of nutrients, of water, of other things to support microbial growth. And so initially, you are at a pretty high level of being aerobic because all the water that you use to put the carbon in the ground, well, it's all has air. So you will start initially in an aerobic state. As as you start breaking down more and more hydrocarbons, you will move to an anaerobic state. And this is normal. Now, it's also quite normal to understand that the subsurface, and this is what I was emphasizing, like an inch is like 13 miles away, that the subsurface, even though your well says it's anaerobic, the subsurface still has aerobic pockets. And those pockets will change as the weather changes, as the season changes. But as a general rule of thumb, the more hydrocarbons you break down, the more likely you are to head to the anaerobic state and be primarily in that state. And that state actually, in the past, was always thought of to be a lot less efficient than the aerobic state because there's a lot more, it's a lot more conductive. You spin the water wheel faster if you dump the electrons to oxygen, right? But if you build a deeper, richer anaerobic system, you actually have a lot more places, in a sense, to dump electrons, and you can end up spinning the wheel pretty much as fast.
Okay. So this question is, what documentation do you have that your additions of microbes survive the mixing and injection emplacement process?
Yeah, we have done quite a bit of genomic sequencing. I've presented some of that data. The difficulty is, and I hope everybody appreciates this, that the microbes we use, are part of who we are as a company. So, you don't really want to tell people, well, these are the microbes we put in, because then you're giving away part of what is your business. And that's different than in a research lab. In a research lab, their job is to give away their business, right? So it kind of, can I say it this way? Maybe I shouldn't. It kind of irritates me because I want to tell people, but at the same time, I know that, from a business perspective, we can't really tell you exactly what microbes are there. So do I have proof? Yes. Can I present that proof? So far, no, that we don't want to do that. But I will be more than happy to tell you what I've seen, and other people can conduct their own experiments and they can publish all they want. And that is exactly what I told you earlier. Some of the microbes live, some of them don't. Which ones make it and which ones don't vary from site to site. And sometimes you think you knocked one out, and later on you'll see it reappear. Now, that is based on its sequence. We don't actually know that it wasn't there and hiding apart from our providing it in some cases. But that's the way I'd look at the microbes. And I apologize to everybody that I can't just put it up on the screen and show you which ones are making it and which ones are not.
Well, on that topic of survival, can you explain how activated carbon shields the bacteria from contaminant toxicity?
The activated carbon absorbs the contaminants and lowers the concentration that is in the aqueous phase. And by doing that, most of the microbes that are most active are those that are on the surface of the carbon or biofilm that is closest to what you would think of as being the aqueous phase. It's not like water doesn't go in the carbon too, but you know, the more active aqueous phase. So that does shield the microbes from the toxic effects.
Okay, and then there's a follow-up question from someone else about this topic that you were addressing in the prior question. If you do lose bacteria species, does the community replace it? For example, next one up?
I think that and I think that this is well indicated in multiple publications, that you're not really building a community based on its members. You're building a community based on its metabolic capacities. So, you know, the way to think about it, if, you know, this is terrible, but if your insurance guy decides to move away and you get another insurance guy, you don't really care that much, right? And the microbes are similar to that in their metabolic capacity. A lot of times one acetogen can be replaced by another acetogen. One methanogen can be replaced by another methanogen. Same way in the hydrolysis scheme. The hydrolysis scheme varies, or those microbes that are breaking down the hydrocarbons, they can vary wildly over time at the site. But if you look at it as overall metabolic picture, what's being accomplished, you see that the parts appear to be somewhat interchangeable.
Okay, another follow-up here. So how do you prove the mass balance of contaminant loss versus simply sorption to the GAC? Is it only from groundwater concentrations, or have you demonstrated that high contaminant mass product is not in the activated carbon?
Yes, we have, and there's another presentation on that, and it's something I intend to publish before long. I just not getting around to it. We have a site called Gossers, and this is just one example. We have other examples, but it's a good one, in which 18 months, you know, before injection, we took core samples, 100 core samples, actually over 100 and something, whatever. I'm not sure what the number was, and then 18 months after injection, we took more core samples. And from those samples, we pulled over 100 laboratory samples. And you can see for every BTEX constituent plus naphthalene, the concentration was lower. That's probably the best way to demonstrate that you are having an impact that is not merely lowering of the concentration in the aqueous phase, that you are not just lowering the concentration in the aqueous phase, you are lowering the concentration in the formation itself. Now, from laboratory studies, we have plenty of studies where we've run degradation to the end, and then we've looked at the carbon itself. But I think that looking at the media and the formation is far more important than just looking at the carbon itself. So I like Gossers as a good example to answer the question you asked with kind of a side swipe. If you want to answer it directly, I would have to show you laboratory data, which I believe we have presentations on that also.
Yeah, it is an excellent question. I think that crosses the minds
It
of
is.
many.
It's. Yeah. I'm sorry, Ed. I didn't.
Yeah, no, go ahead.
It is. It is. And in fact, when I first saw the BOSS products, I thought, wow, because I was, you know, trained as a risk assessor and a toxicologist initially, and I thought, well, this is great. You change the entire risk scenario, you get the carbon in the ground, it absorbs it up, and later on microbes will finally get around to it. Little did I realize how active a microbial community could be built. And there were some other people who said, no, all you're doing is just sorbing it to the carbon. And that was one of the inspirations behind, say, the Gossers study and some other analysis that I did early on, long before I ever worked for this company, because we were spending state funds. And I'm thinking, Jesus, I can't leave. Oh, I'm not supposed to say that online. But I was thinking, I just can't leave, contamination bound up in this stuff everywhere. I got to figure it out. So yeah, we did quite a bit of work to make sure that we felt comfortable that what we were doing was the right thing for the people, at least at that time, for the people of the Commonwealth.
Right, which, the very nature of this process is we're putting a platform for treatment in the ground. And, trapandtreat.com, RPI's website, really trying to emphasize this, that we're not just soaking it up, we are creating the opportunity for treatment to occur at an accelerated rate. Looks like we're at the end of the questions that are posted. Thank you very much, folks, for your time and for posting these questions. Really, really good stuff. And I'm sure other things will come to mind for you. Feel free to reach out to Dr. Winter anytime. We don't have a slide on here, I guess, Ed, with your contact information, but I've mentioned several times the website address. You can reach Ed through there, trapandtreat.com. You got a slide that you can pop up, or?
I don't, but it's ed@trapandtreat.com. That can be too hard. It can be too tough.
ed@trapandtreat.com is the email. And I can be reached at edcreedon@astenv.com. But better yet, you can go to the AST website, astenv.com, and we have a form for contacting us. You can reach out to us there and post some project information. We can help you determine if these technologies are the best remedy available for your site. We certainly do that at no cost. And we really do appreciate everybody coming on today. This is a pretty heady topic, and maybe as you sit and mull this over, more things come to mind. So reach out to us really at any time. Got some nice thank yous coming in on the Q&A for you, Ed. Great job.
Well, I sure appreciate everybody giving me a piece of their time, and it's been great.
Yeah, okay. So everybody have a great rest of your day. We will be signing off now, and watch for the recordings. They will show up in your inbox on an email, and you can always go to those websites that you surely have memorized by now and find recorded webinars there. So thanks, folks.